So we're now on mysterious moving molecules. So you guys will be looking at that lab, and part of that lab is a data chart, and on that particular data chart it says fill in wells 1 through 8 with the name that is on the side of your container. So in front of you I have 8 little tubes that are filled with solutions that we're going to be filling in 1 through 8 on my gel electrophoresis. First thing you're going to notice is a little thing from spectrophotometry. My colors are Roy G. Biv. So we have red, orange, yellow, green, blue, but then we also have a couple unknowns there. You're not labeling them red, yellow, green, blue, so on. There are names. So well number 1, I'm going to kind of hold the tube up so you guys have to go through the video to get the names on them because they're not called red and orange. So well number 1 is going to be this solution. So it's pyridine Y is what's going to be in well number 1. Well 2 is methyl orange. Well 3 and well number 4 with the green cap. Well number 5, which is the blue cap. Well number 6. These are just listed as unknowns. So well number 6 is going to be unknown number 1. Well number 7 is unknown number 2. And well number 8 is going to be unknown number 3. So once again, we have 5 with names. That's my red, orange, yellow, green, blue capped ones that I just showed you. Don't list them as red, orange, yellow, green, blue. If you need to go back through the videos to get the spelling, I just held them up in front of the camera for you guys. So now we need to load those solutions. We're loading 8 microliters into our gel. So on this particular gel, it's a little different than the practice loading gels that you guys saw in the last video. I only have wells in the middle here. So I have mid-load wells. That's because I'm not sure the charges of the molecules I'm going to be putting in here. So remember, gel electrophoresis separates out molecules based on charge as well as molecular mass. You can see here, this is the black end of the box. This is my cathode. This is the red end of the box. This is my anode. So when we load things into the middle, and then we have this buffer inside, which is kind of like a electrolyte solution. It's kind of like a Gatorade. It's going to help to conduct the current. And we put that charge through there, molecules are going to move based on their charges. So if something, because opposites are going to attract, if something is a positively charged molecule, it's going to move towards my negative cathode. So cations, which are positive, think about the T in cations, positive, are going to move towards the negative cathode. Over here, anions, think about the N in anions as negative. So my anions are moving towards my positive cathode. Opposites are going to attract. So I'm going to have molecules going on both ways depending upon their charges. Next, we're going to look at their molecular mass. So in terms of molecular mass, what we're going to see is that things that stay closer to the origin, same thing as chromatography, here my origin is my wells. That's where we started from. And my solvent front is going to be from the wells to the end of the gel. So before we measured the paper from where it was like wet versus dry, here we're going to measure from the wells to the end of the gel. One thing for you, when you go to measure solvent front on your diagram, you need to measure both directions, because depending upon how this comb was put in, you'll see it has kind of like a little shelf there. One way is going to be longer by about a half an inch or so than the other way. Okay, so you need to measure solvent front towards the cathode and solvent front towards the anode, and they're going to be different distances for you. Okay, so I marked my number 1 well because my top well is going to be at the top here. Number 8 well is going to be down the bottom here. I'm going to take my first one. Once again, here's my solution for my first well. I'm going to measure 8 microliters. These little solutions have already been vortex, so they've been mixed up. And I am measuring 8 microliters of it. So once again, this is called submarine loading. I'm gonna try the best I can around this camera here, but we're gonna do the process of submarine loading. That means the tip of my pipette is gonna go into the buffer there, and this material that we're loading into the gel is heavier than the buffer that it's in. So it is going to sink as long as we get it close to that well. And like I said, this is a little difficult for me because I'm not used to doing it left-handed and doing it around the camera, but we're gonna go nice and slow. And load that first well. So you can see that first well is now loaded. We are going, changing tips. We are going now load our second sample. Let me recap my red one, and now I'm going to load my second sample. My second sample was that methyl orange. This is going to go into well number 2. I might switch directions on you guys here and try to do it right-handed from the other side now. Let's see what I can do. So we're going to go through, get it close to my well, and slowly release my sample. It is a slow process loading them up. So well number 2 is now full. Well number 3, you're going to notice that all 8 of our wells are going to get filled here. Well number 3 was our yellow solution. Same thing, fresh tip, 8 microliters of that particular solution. I'm only going down the first push on the micropipettes. Remember, it's first push to aspirate up. It is second push to release or deliver. So now I'm going into well number 3. And it's much easier to see these little wells when you guys are directly over top of them. So if you're going, where the heck is she putting them into? There are little openings there that you can see. They're just easier to see. So you might be able to see them down here on the bottom. I'm going to slide this gel over just a little bit. Those little dashes, that's what I'm looking at. And they're a lot easier to see when you're directly over top of it. That was the third one. My fourth one is my green solution. That is my green one here. Same thing, fresh tip, 8 microliters, first push to aspirate up, second push to release. Cap that, push back, here I go into my fourth well. And so as soon as I start releasing stuff, it's going to sink. You're going to notice sometimes some of your excess stuff over here might get stuck up there. I'm just kind of dissipating it within the solution. But that substance is now stuck within the gel. It's going to travel through the gel, not on top, not underneath. It's going to travel through. Same thing like your chromatography paper. This gel, this sugar structure here, is your matrix. It's what the molecules move through. So in chromatography, your solution of marker and water went through the paper. Here, it's going to be going through the sugar gel itself, that agarose gel. We are now on well number 5. Here's well number 5. 5 was our blue solution. Now I have my 3 unknowns. So as I go through, I have unknown number 1 is going to go into that 6th well. I'm going to cap him back up. Let's load that 6th well. So I want to make sure I get everything into that well so I don't cross-contaminate. If I hold this pipette too high and drop it off into the buffer, it's going to kind of sink to whatever well is closest. Now an unknown number 2. So now we have unknown number 2. It's going to be going into the 7th well. Now you're going to notice the colors that are going in there, they're not matching the caps of the bottle you had. So where we said, oh, we have Roy G. Biv, they're really not going to match, as you guys see, which is why I don't want you just to label them as colors. They actually have names, except for your last 3, which are your unknowns. There's well number 7. And I have my unknown number 3, my last one that I have here. And I'm going to load well number 8, the best that I can around this camera. It's a slow process. First push, second push, pull out, and I'm releasing. That's it. Micropipette goes back on the rack. So now what you guys see here, I'm going to kind of slide it off that red line, that gel, so you can actually see the colors that we have within there. So those are now stuck. They're not able to move. They're stuck in those little holes that we have there. I've put online for you guys a Word document that shows my initials. So you guys are going to go through initial and you're going to color in those wells. Well 1, well 8, black side, red side, cathode, anode. So now we're going to go through and we're going to hook it up. So when we hook it up to our voltage box, we're going to have cords, it's kind of like a car battery, and we're going to hook up our anodes and our cathodes. So we have our little voltage box here within the room. We actually have a whole bunch of them. We're going to kind of expand out here in a moment. Let me plug everything in for you first. And we're going to have some cords that go along with it. We need to run electricity into this particular box that we see here in front of us, because we said that's how things are going to be able to move. So I'm going to kind of expand this camera. Hang with me here so you guys can get a little better view of what's going on. And let me back you up a little bit. And there's the rest of it. So off to the side here, you see my little voltage box. I'll slide it a little closer. We have 2 spots for 2 different voltage boxes to attach to. The lid needs to be closed. My black side, I have my black wires. It goes right into the side of the box. Red side, same thing, goes right into the side of the box. Red goes to red, black goes to black. Now I need to take these 2 wires, and I have the other end with the probes on them. Those probes are going to go right into the box. They're staying on the same side. Red goes to red. Slide them in. Black goes to black. Now we can kind of turn it on. And my numbers are now going to pop up. It's going to tell me what my voltage is in there, my millivolts. And it's set to about 97 right now. I'm going to leave it at 97, which is a little higher than I like, but it's a shorter class period. That's going to run. You guys will then see a 10-minute picture on the Word document. I'm not running this for 10 minutes. I'm going to do it now, and then I'm going to show you the end one here in a moment on how you guys will calculate things out. So you will see a picture at 10 minutes. What does it look like? You're giving me a sketch of that 10-minute picture that you see there. Mark your black end, mark your red end. You're going to see molecules moving in both directions. So as that's running, now we can go through, let's say we're completely at the end and we want to kind of go through and break down our particular molecules. So I have another setup. I'm going to take it out. I'm taking my gel out of the box. My buffer in there is going to be a little bit warm because we have electricity running through there. And what I can now do is I can take it right from the solution and I can kind of slide it right onto paper so that it's easier to see. Those colors that you see there are stuck. I'm going to blow it up here for you in a moment. I know it's a little glossy and got some other stuff going on with it because it's still wet. And you guys will see a final picture as well on your thing. But that's what we have. Got some glare on it, so I'm going to try to hold it up. There is my black end, my red end. Well 1 is at the top, well 8 is at the bottom. So right now I can tell you whatever was in well 1, we did not have any separation. Meaning, think of well 1 as a single strip of chromatography paper. So well 1 is its own little chromatography paper there. Hold some of this stuff up for you guys so we're not glaring here. Okay. There is no separation. It's kind of like if the black dot just ran right along the chromatography paper. But what I can tell about that particular molecule is its charge. I can also say it's about medium weight in terms of its molecular mass. So its charge is going to be positive, and I know that because it's going towards the negative cathode. That means that this molecule is a cation. Let's go to well number 8. Did I have separation? Yeah, my unknown number 3 separated into a blue molecule, and then it also separated into a very faint pink molecule. That means I have to calculate out 2 measurements in centimeters. I have to calculate out my RF values. I have 2 of them. I have 1 for the blue molecule, and I have 1 for the pink molecule. And you go, how do I calculate out RF again? Oh yeah, distance the molecule moved, divided by my solvent front. So where this comes in a little play, so now we have to think about how am I going to put rulers down? And you will see pictures that look like this as well. So you're going to see one that looks like this, and I'm going to have the ruler starting at the origin and going all the way over. And then I'm going to have one going the other way as well at the top. And the reason is because I want to see the distances from the anode as well as the cathode. You can tell this solvent front is longer than this solvent front. That's why you need to measure in both directions. So as you guys go through, you will go through and measure it. If you're measuring off of here, so let's say if I'm measuring this little red band here, it is a band. Let's go with this one. It's a little brighter for you guys to see. My advice, I would go to the middle of that band. You can say, oh, I'm going to go to the beginning of the band, I'm going to go to the end of the band, or I'm going to measure in the middle of the band because it's kind of a blob. However you do it, keep it consistent for all of your bands. Okay, keep it consistent there. My next advice, if you're doing this on camera, meaning you're watching this video or you're doing it off the pictures that I put there for you, if you have a piece of white paper at home, hold it up to the screen. Hey, I want to go through and I just want to look at well number 8. There's well 8. Or take 2 pieces. Hey, there's well number 7. Yours is going to be a little easier to see. I have a lot of glare on mine right now. The pictures you see online will not have glare. Once again, this does not wipe off. It is stuck in the gel. So if I make that gel thinner, less sugar solution in it, those molecules will run faster. Here's the next thing. If I let this keep running, let's say I forget about it and I leave it on overnight, I come in the next day, I'm like, oh, I'll just check my gel then. There's not going to be anything on here. There's nothing that stops them from falling off the edge. They would fall off the edge, they would go into solution, and I would kind of lose all of my data. So this is a timed event. We want to make sure things don't go too far off and we actually lose our particular pieces there. Okay, so that's what we're looking at. You guys will see a couple sketches online. You guys will see an initial sketch photo that I took, and then you'll see a second one at the initial where I kind of slid off the red line so that you guys could see the colors better. I have a 10-minute picture for you, and then I also have a final picture for you with the rulers there. Do not use this one. It's hard to see with the glare on. I'll make it into a Word document so you guys can pull it right off there and look at it. Okay. Every well, like I said, has its own separation. So on your chart that I gave you, it's going to say, oh, do you see separation? So I can say, oh, for this particular well, the 7th one, yes, there's separation. I see yellow, I see purple, and I see red. I see 3 molecules that broke out of that mystery molecule number 2. I have 3 measurements, one for yellow, one for purple, one for red. I have 3 RF values, one for yellow, one for purple, one for red. I can also say that I can tell the charge of them all. I can tell which one's heavier than the other. I can say molecular mass-wise, that red one's pretty light compared to the yellow one. I can also say those molecules are also all negatively charged because they're all moving towards my positive anode. Okay, so go through that, look at the Word document, use the information and the data from the document rather than from this video. The only thing you're getting from the video is the name of what's going into each well. And then you'll do your measurements right off the document that you have there. And then let me know if you guys have any questions when you get to the questions in that particular lab. Thanks.